My name is Marc Streit. I'm a full professor at Johannes Kepler University in Linz, Austria. And the title of my presentation is Injecting Life into Visualizations, which could be pretty much anything, right? And before I'm going to start my talk, I will briefly introduce myself. So I did my PhD at Graz University of Technology in Austria, and then moved on to become an assistant professor at JKU Linz. And during my tenure track, I spent some time at Harvard Medical School at the bioinformatics department, and also later at Harvard School of Engineering as a visitor professor on a Fulbright scholarship. And then I'm still teaching at Imperial College in London, and I'm also the CEO of Datavision, which is a spin-off company that develops data visualization solutions for pharmaceutical and biomedical R&D applications. All right, so solving complex domain problems requires a mix of different disciplines and skills. You're all fully aware of that. So it requires data wrangling, databases, data management, software engineering skills, machine learning skills, statistics skills. Understanding the domain problem is, of course, super relevant. And visualization also became an important part of this data science toolbox. So the question is, what can visualization actually contribute? And visualization is basically the process of transforming data into static or interactive visual representations. And you might have heard the quote by Richard Hemming, who said, "The purpose of computing is insight, not numbers." And if you apply that to visualization, it would mean the purpose of visualization is to gain new insights and not to create pretty pictures. So creating visualizations and using visualizations goes much beyond just creating visually appealing representations. And if you look closer, visualization can be used for different purposes: to present data, to explore data, to confirm findings, and also to explain something like a complex model, for instance. And let me start by talking about presentation. So this is probably the most widely used purpose when people think of visualization. And if you look at Watson and Crick here, they use this nice hardware model of the DNA double helix, of course, to present what they already knew. So they didn't make the discovery using this hardware model, which is also some kind of visual representation, a visualization, but they just tried to communicate what they found. And this is this whole purpose of using visualization for presentation. So visualization is really omnipresent today, and you will see lots of bad visualizations also out there. There are great examples, for instance, this nice line chart with the data hidden in the leg of this nice lady. Then also, that's probably one of my favorite examples. It was tweeted by BBC, and they just tried to come up with a ranking of the most played artists in their Festive 50 show. And they had this great interactive pie chart where you're able to scroll through 11 pages of little chunks of the pie. And of course, it's impossible to associate the colors with the chunks of the pie. And for instance, this one that became famous as the banana chart. And there's also a nice platform. It's called WTFvisualizations.net. And it's a really great resource where people are collecting those examples. But also on a more serious note, if you look at scientific papers, there you will find cluttered visualizations, and you will find many visualizations that don't follow the standards and rules the visualization community has come up with. Let me give you one example. And many people still think that 3D is sexy. If you look at this banana bar chart, of course, you will immediately see that this is unnecessarily complex. But those are just one example. But people still like to use 3D. And some advice would be avoid unjustified 3D. And I also have a nice example for you here. It's this 3D bar chart. And if I ask you which one is the tallest bar, it will probably be not too easy for you to find the answer to that question, because it could be this one, it could be that one, it could be that one, right? Also, answering the question, what is the pattern in the data, is not that straightforward. But it's not because the pattern is so difficult, but it's because the visualization, the complexity of the visualization is actually hiding it. If you would pick a simple heat map here, it would become very obvious that this is a question mark. And the reason is because this 3D, you have to deal with occlusion, so bars hide each other. You have to deal with something called perspective foreshortening. So if things are further back in the visualization, they will appear smaller, which has nothing to do with the data. And of course, there's an exception to this, which is if you visualize actual 3D phenomena. And one example would be this protein structure here. And of course, there it makes sense to work with occlusion, to work with size. So if things are closer to the observer, they're bigger with shadings, because those are the visual cues that we also use in real life. Another example would be if you have an actual three-dimensional scan of a human body, for instance. That would be another example where you should go for 3D, of course. And there is a nice series of columns with, like a nature method column. There are actually two. One is called points of significance and one is called points of view. So points of significance is still active, points of view is not active anymore. But the idea was that in the case of points of view, visualization researchers are trying to communicate the really fundamental things about how to visualize certain kinds of data. And the same is true with points of significance, which is about basic statistical knowledge. And I had the chance to contribute to two of those columns together with Niels Geomborg from Harvard Medical School. We, for instance, wrote an article about box plots and bar charts, and another one about how to visualize time series data. And you see it's really quite basic stuff, but there is also there are quite some rules that you can follow in order to make your life easier. And I think it's also important to educate other communities how to create effective visualizations. So this was everything about using visualization for presentation purposes. But there is also the whole world of exploration. Interactive data exploration is much more interesting for research because it's about extracting new insights from your data. And now you could say, well, cannot we just use automated techniques to analyze the data? Why do we need the human to be part of this? And this goes back to John Tukey and others. And in particular, John Tukey coined a term called exploratory data analysis. And a quote by him is, the greatest value of a picture is when it forces us to notice what we never expected to see. And this is so true about visualization. And nowadays, those fields are called visual analytics and visual data science. And it's basically about combining algorithms, automated techniques, modern artificial intelligence models, for instance, with interactive visualization. So it's about this back and forth between humans and the machine. And I want to show you some of our work on exploratory data analysis. There are many examples that I would love to show you. But I will start with the result of a multi-year collaboration with the German pharmaceutical company Boehringer Ingelheim. And together with bioinformaticians working at Boehringer, we developed a web-based tool that helps researchers to identify and characterize potential drug targets. The tool is called Arduino. It was published as an applications node in bioinformatics. And it's designed to let users interactively rank genes, tissues, cell lines, and other biological entities. The tool is also publicly available as open source. So the workflow in Arduino is quite simple. you start with a list of items. Those items could be genes, cell lines, tissues, indications, patterns, depending on the data you have at hand and the data you have stored. And the system comes with a couple of public data sets, but you could also roll it out internally with your own data. And then this list of items, so it could be a set of known cancer genes, it could be TCGA cell lines, and so on, serve as an input for the analysis. And then the next task is the interactive exploration task. You can use the system to interactively rank, filter, and select the items that you're interested in. And the last step is then obtaining more details about the item. And the core of Arduino is actually a visualization technique called lineup that we published a couple of years ago. It's specifically designed to create and understand multi-attribute rankings, which are rankings where multiple attributes, multiple dimensions, contribute to the ranking. And you might weight them and combine them in order to get to the items you're interested in. And the technique actually was published as open source by the PhD student working on the project. The student also won the Best Paper Award at the biggest VIS conference. And Microsoft picked up the open source project and integrated the technique as part of their Power BI business intelligence software. And I just want to very briefly show you how users can explore the data using the technique, because you will see that this goes beyond what you know as interactive standard plots that you might be aware of. So for instance, here in this example, and I hope you can see my mouse, but that should be fine, you see a list of 1,000 cell lines. And the cell lines in this case are grouped by tumor type. You see we have 4 different tumor types. And we have additional data available that the user could add as needed. And if I start this little screen recording, you see that the user could further aggregate the data so that you see how they're summarized, what's the distribution. You can dig deeper into one of the groups. You can set filters. You can even go down. So this would be to work with the ranking, and you could even go down to the item level where you can read the different data descriptions. So about 4 years ago, we founded a spin-off company named Datavision, and by we I mean Sam Gratzel, the PhD student who implemented lineup, Niels Gellenborg from Harvard Medical School, and Alex Lex from the University of Utah. And Datavision actually maintains and extends the Arduino application and the target discovery platform sitting beneath. And based on the same platform, Datavision developed a couple of tailored web applications that are now deployed and in active use in various big pharma companies for exploring indications, chemical compounds, patterns, cell lines, genes, and so on. And if you want to know more, I'm happy to get back to you. And by the way, when we reserve a table for company events, this is what we get. So for instance, you see here the reservation then says Tartarvision, Detervision, which means like wiping in German. But I think that's the price to pay for a unique Google hit. All right, so far I talked about presentation and exploration. And another important goal of visualization is to confirm and reproduce findings we made using exploratory data analysis. And this is, as you can imagine, not straightforward. And I want to show you some of our work that is addressing this reproducibility problem. So scientific progress in general relies on the fact that we can trust and reproduce previously published results. But in data-driven sciences, that turned out to be very difficult. So how can you reproduce what you or someone else has found in the data? Well, in other domains, reproducibility is more straightforward than in science. So there is what you see on the photograph here. It's a little village in Austria called Hallstatt. And the funny thing about Hallstatt is that there is a replica in China of this little village. And my question to you would be now, is that Hallstatt that you see on the photo in Austria or China? What do you think? And I guess you can use the chat and Seth will forward that. Or maybe just Seth can try to figure it out. What do you
We have 3 people who have said Austria, 1 who has said China.
Okay, yeah, the Austria gang is actually right. So I think it would be hard to recreate the mountains and the large lake. So this was Austria. How about this one, Austria or China? So that's the center of the little village. Let's wait for the vote again.
The consensus appears to be China on this one.
No, that's still Austria. I have a third one, and now it would be quite obvious that this is actually China. But why I've added this example to my slides is because if you reproduce Hallstatt, this little village, it only needs a blueprint or photos. But what about findings from data-driven sciences like biology and medicine? There, it's not so straightforward. So let's, for instance, look at cancer research again. Amgen tried to reproduce the findings reported in 53 high-profile cancer genomic studies. And it was not possible to confirm of like 90% of the findings of these studies. And that at that time, that shook up the whole community. So one key to reproducibility is provenance information, which means that you keep track of every detail, which of course is it's a quite difficult problem, as you can imagine, because it goes through the wet lab, it goes through all phases of the project. So computer science can only like solve a part of this problem. But I want to make the point here what you can do if you try to reproduce what you have found using visual data exploration. And this is an example I've picked. It's one of our own work. So a couple of years ago, we published a visualization, which we called Stratomax. This is the visualization here. It was created, we designed it for exploring genomic stratifications in cancer, at that time, TCGA data. And it was published in Nature Methods. What I want to say here is that if you publish findings that you make using a visual tool, you use a series of screenshots or maybe also a video. We did both. And these screenshots, they come then with long descriptions what the user actually did. But of course, this is very limited because images are insufficient for telling the story of visual discovery because they lack the full provenance information and context. Videos are also difficult to produce and edit. So both techniques are not the solution. And you, of course, have the problem that you cannot answer what-if questions. So if someone would say, like, what would happen if you click in the software on this other band here appearing in the visualization, or if you set a filter in a slightly different way? Well, this is something you cannot answer. And if we look at the problem, you have those two worlds. We have exploration and we have presentation. And usually people start with exploring the data using interactive tools, but could also be by other means. And then you switch to presentation. That could be that you just add a screenshot to your PowerPoint presentation and you're then presenting your finding to someone. The problem with this process is that if you add a screenshot to a presentation or to maybe a paper, it's very hard to get back to the exploration and to reproduce the visual discovery that you made, but also to follow up on what-if questions. And if we look at this on a more abstract way, you see that if people start with exploration and then switch to what I call here authoring, that would be you made a finding and then you try to somehow like persist this finding using a PowerPoint made by making a video, and then you're going to present this artifact, this video or this figure in a paper, in a presentation or whatever. And with this work, we did a couple of years ago, we actually tried to close this loop. We called the system histories, where users should be able to go back from presentation to exploration again. And this might sound quite abstract, but I will try to make it more concrete with an example. So in the paper, we actually demonstrated our method by two examples. One was Gapminder, so it's a little tool for exploring data about countries. It's basically a scatter plot where you have a timeline going through the different years, and our own Stratomax tool. And we wanted to show that it's easier to reproduce findings and to answer those what-if questions. So let me show you this Gapminder example, because it's much easier. I don't need to explain the whole genomic stratification stuff. So in Gapminder, I will start the screen recording. The user can scroll through time, and you see that you have every dot is every point is a country, and you see here that we have life expectancy versus GDP mapped on the axis. And in the system, every step is tracked that the user is doing, but not in a linear fashion, but you could also go back and introduce branches. So it's more than just a flat history. You're creating a history graph. And this is one important aspect, because this kind of history graph then allows you to use this captured information for storytelling. And what the user then can do is, oh, sorry, that was one slide too far. The user can then switch to authoring, and this works like a video editor. So you can pick the steps from your analysis that you've done live before, which you want to make part of your story that you want to share and share with others. And you can click through the steps, and the system will try to use animated transitions to explain what the user did between the steps. And you can also add annotations. Well, this is what you would also do using PowerPoint, right? You would just create a little arrow and say, well, this is Somalia, and Somalia is relevant because of something. And then you can go through the story, and you can also change the timing if you want to like tell the story in an automated way to auto-advance. But now if you go to the third mode, which is presentation, you can share the link to the web application to someone, and this person can, in a read-only fashion, just scroll or step through the different steps, which are basically the story of the visual discovery you made. So you cannot just share the finding, but you can also share how you got to the finding. And you could even go one step further. You could switch back to exploration and try something else and create an alternative path or branch in your history that you're going to share with colleagues again. So that's how we imagined histories to work. And the goal was really to make exploratory data analysis more efficient, more collaborative, and in the end also more sustainable. And this idea could be applied to many other problems and data sets. So for instance, collaborators from University of Groningen, they applied it to radiology data in the context of decision-making in medicine. So if they had collaborators at a clinic which do those tumor board meetings to discuss the treatment of patients. And there they could prepare different stories, different alternatives, and go through them and share them with the rest of the team, for instance. And vStories is now also fully integrated into the Arduino visual analysis tool for drug target discovery that I've shown to you, that is rolled out at Boehringer, at Bayer, for instance. And you see that there is this history, and you also see that there are alternative branches, for instance, that keep maybe dead ends that you hit in your exploration. By the way, related to provenance, we are also currently working on a tool for visualizing patient cohorts. And the tool supports users in splitting, filtering, and characterizing cohorts based on data from various sources. So the graph here that you see is interactively created by what the user is using for filtering and subsetting the cohorts. And then you can, for instance, compare different cohorts with each other and try to characterize them in further steps. And this is now, it was very recently accepted to bioinformatics as well, and it's the preprint is online already. Okay, so that was this confirmation aspect where we really try to use visualization for confirming and for reproducing patterns that you made using visualization tools. And in the last part of my talk, I want to talk about how to use visualization for explaining the functioning of complex algorithms, which could be any kind of algorithm you may think of, but also like a deep neural network. And I will show you some examples. So this goes into the direction of explainability, of explainable AI, sorry. So you all know, I guess, that high-dimensional data plays an important role in many scientific fields nowadays: biology, medicine, physics, and so on. And a very common approach to deal with this kind of data is to use dimensionality reduction techniques such as t-SNE or Umap. And the goal is always to reveal similarity and patterns in the data by reducing it to 2D or 3D. And the closer 2 points are here, for instance, in the scatter plot, that's the famous iris data set, and you just reduce it to a lower dimensional space. The closer 2 points are, the more similar they are in high dimensional space. So that's the assumption and how to read it. People are using embeddings, could be static ones, could be interactive ones in various kind of contexts. So here would be a simple embedding of old books on Wikipedia, but here would be a more serious examples showing an embedding of breast immune cells. And you might have, there was a huge discussion also on Twitter about the value of embeddings and what could go wrong if you interpret the patterns that you see in those embeddings. And you should be really careful when you interpret those patterns. But I think the techniques are still useful, of course, because sometimes there is no other way around interpreting the high-dimensional data sets. But of course, you need to be very careful. And there's, for instance, a cool Distill article on how to use t-SNE effectively. It nicely guides you through examples of what can go wrong when interpreting such embeddings. For instance, hyperparameters really make a difference. The cluster size that you see, so clusters that appear might mean nothing, the size. Distances between clusters might not mean a lot. Random noise could not look random. So there are a couple of things that you need to keep in mind when you do that. But in the line of work that I'm going to show you, we exactly try to provide additional visual means to help people understand this high-dimensional data that is projected into lower dimensional space. This would be another article on understanding Umap, which is also nice. You can play around with different toy data sets and set the different hyperparameters and see how the results change, and the change in results is really dramatic sometimes. This would be a cool example of a Umap projections of a 3D woolly mammoth skeleton. So this is the actual three-dimensional data that is down projected to two-dimensional space. And you can see, for instance, that the structures are well preserved. And in this on this interactive article, you can also change the parameters and see that it will preserve different aspects of the data, of the actual data patterns. And I now want to show you some of our recent work related to those embeddings. And we looked at the problem of visualizing sequential data in the context of embeddings. What do I mean by that? So there is a important related work by Benjamin Bach and colleagues, and they published a technique that is called TimeCurves. And the fundamental idea is that you fold time to visualize patterns of temporal evolutions in your data. So imagine you just have a sequential data that could be a Wikipedia article, for instance. Okay, and you have a timeline. So the Wikipedia article was changed multiple times. And then the idea is, instead of having a straight timeline, so the distance is basically how much has changed between the articles. And then you're changing the shape of the line in order to push similar articles towards each other and to push or to put them closer to each other and push dissimilar articles away of each other. And then you see that you get those time curves. And if I show you examples, it's a very simple but very effective technique, I guess. So this would be applied to surveillance videos of a street. So you have a camera pointing towards the street. If nothing happens, this is basically this blob of points. Every point is a video frame. And if someone is walking through this video, then you see that you have those. Of course, they are more dissimilar to the images where nothing happens, and then you see those patterns appear. This would be another example from the paper. It's showing different Wikipedia articles, the time curves, and you see that, for instance, abortion, it has started here, and you see that, for instance, there was an edit war going back and forth with huge changes. Someone was changing it, someone was changing it back. You also see that, for instance, that's what happened with Ukraine. So you have lots of little changes, and then all of a sudden there were huge changes back and forth, right? And this is something you can effectively see using those time curves. And also in the paper by Bach, I found it really interesting because they try to characterize the patterns that you can see. For instance, you have cyclic patterns, you have outliers, you have oscillation, like with this abortion article, you have alternation, you have transitions, and so on. And they also looked for geometric characteristics, like self-intersections and so on. So now, why is this relevant for my work? And I want to show you an example, and then I'm moving on to more serious examples from cancer research and so on. But I want to start with this Rubiks Cube. And we got inspired by the time curve technique. And I decided to look for a student who is interested in solving Rubiks Cubes. And as you can imagine, there was a student who is interested in solving Rubiks Cubes. And I'm sure most of you had held a Rubik cube in their hand at some point. I don't know if someone managed to solve it properly, maybe with a manual, maybe alone. But there are even tournaments for solving Rubik's cubes. For instance, the world record, or at least when I looked it up, was 4.22 seconds. That was the world record a human used to solve the Rubik's Cube, and the world record of machines was at that time 0.38 seconds. So I have also a video embedded here. So that's pretty impressive, I guess. So how it works is they all get the same scrambled cube, and they're allowed to look at it, and then they need to put their hands down, and then the time starts running. And below you can see this video of this machine-solved Rubik's Cube, which is also cool because they actually had to manufacture a new Rubik's Cube that actually holds for these kind of examples, because a regular Rubik's Cube would be destroyed immediately. Okay, so and the goal of this student was actually to come up with a visualization that is telling how people or how algorithms solve Rubik's Cubes. Okay, what are the strategies applied? And this is the result here. And I will go into details of how to interpret that. On the left-hand side, you basically see the visualization of 100 Rubik's Cubes and how they were solved using a method that is called the beginner's method. So that's the method you would probably use, or I was using. And then on the right-hand side, it's a more a faster way to solve the Rubik's Cube. It's called the Friedrich Method. And before I'm going into details of how to interpret the patterns that show up, if you take the Rubik's Cube, what we did is that you virtually unfold it, okay, and you just see it as a vector of features, which are the colored cubes, and then you put it in a projection. You create an embedding where every state of the cube is one point, and when you make a rotation, there is another point and another point, and you just connect them. If you do that for one cube, you get a result like this, and you see that there are those curls appearing. And this is basically, if you've tried that, if in order to get one piece to a different position, sometimes in the end, you need to make 10 rotations in order to get it done. And these are those curls that appear. And if you do that for 100 cubes, you see that there are patterns, patterns appear. And this is another way of explaining it. So you have a Rubik's Cube, it starts scrambled, then it's solved iteratively. You see those as feature vectors that you put in a projection, and you're just connecting those. And you get a path going from the start to the solved Rubik's Cube, which is the star here. If you do that for multiple ones, you see that the starting points are scattered because they are scrambled. And then they will all approach the same final state, right? Because they're all solved in the end. This is an example of the same Rubik's Cube solved with those two strategies. And you see that first, in the first like half of the solving, it shares the path. And then those algorithms really work in a different manner. And you see the beginner's method is forming those curls, which is pretty time consuming. And the Friedrich method is more defined. So it's quicker in solving the Rubik's Cube. And if we now look at this result, you see that what is marked with 1 here is all scrambled Rubik's Cube, which is the beginner states. And then they all meet here at the solved Rubik's Cube. And what is interesting with the beginner's method is, or in both methods, this cluster here annotated with 3 is cubes that are solved except one piece, okay? And in order to get from here to here, you need to go through crazy rotations. And this is why the beginner's method also takes more time, because the Friedrich method has a shape that is more defined, that you see here, that is also shorter and takes less rotations. But of course, it's not that intuitive. That's why people are not using it if they're not experienced solvers. And this is the tool we call Projection Space Explorer. And we published it with various examples. And I just want to show you how to interact with the tool. And I hope you can see it. Let me, now you should be able to see it. Is that correct, Seth? But I will just continue.
It is. Yeah, we can see it good.
Okay, perfect. Thank you. I will just make it a little bit bigger. And now I will go to this example with this Rubik's Cube that I've shown to you before. And you can then, as a user, start exploring the patterns. So for instance, if I hover over it, you see those are the scrambled states here initially. If I go closer towards this star shape here, you see those are the almost solved ones, including the solved ones, right? And then the tool also allows you to, I will just make it a little bit smaller again, so that the patterns look better. You could then, for instance, go and investigate the different parts. And if you make a lasso selection, you will see, for instance, that all states that are part of this lasso selection have this part already solved. And where you see gray, it means that there is some variance in the cluster. And if I make a selection that is closer to the solved state, you see that almost all phases are already solved except the top layer. Okay, that's how to work with this tool here. I will just close the demo again. Go back to my presentation. So if you want, you can just play with different data sets. It's pretty fun if you know the problem. And we also, or the student, created a demo for an open house day, which was pretty cool. So we had a Rubiks Cube that was tracked via Bluetooth. You can buy that on Amazon. And people had to solve the Rubiks Cube, and we showed instructions on the screen how to what to do next, because many people cannot just solve a Rubiks Cube. And this was an early version of the visualization showing up on the screen. And if, like the visitors of this exhibition, if they gave up, they were just able to put the cube into this Lego Mindstorm solver, and the Mindstorm solver solves it until the very end. So that was this open house day exhibition. And we don't need to watch it until the end, but here you see that it's actually doing pretty good job in solving the cube. All right, and the student actually also implemented a multiplayer mode. So we had 2 Rubik's Cubes that were Bluetooth tracked, and 2 visitors were able to actually compete against each other to solve the Rubik's Cube faster. And you see those were the initial, like the earlier versions of the visualizations, which don't look that nice. But we also applied it to various kinds of algorithms. So basically, it could be applied to everything that could be seen as a sequence of high-dimensional data. And here you see a comparison of different sorting algorithms, like bubble sort and quicksort. And you also see that there is quite some difference in the projections if you apply that. So this would be Umap, T-SNE, ISOMAP. And I think the learning was that there is no best projection. Sometimes one projection shows up some aspects of the algorithm, and sometimes another one shows different aspects. So there is no best solution, I would say. We also applied it to chess games, for instance. I have a colleague sitting across the hall who is also a professional chess player, besides being a theoretical computer science professor. And he's looking or interpreting the projections with us. And you see, for instance, those are chess games. So they all start at the same chessboard with the same chessboard. And then you have 2 openings. And in that case, it's 450 professional chess games. And you see then there are interesting patterns appearing. So for instance, this cluster E, those are end games. This B and C, those are castling moves, and so on. But you need to be careful with interpreting them, because if you just change the perplexity, which is a hyperparameter of T-SNE, then you will get a very different result. It doesn't mean that the other result is useless, but you just see different patterns. And there is also related work. Researchers, colleagues tried that with for instance, showing the evolution of training a classification network. So they used MNIST as a test data set, and you show that the training epochs are encoded using grayscale, and you see that during the training, the projection is moving those instances, those images, to this outer space in order to differentiate between the different clusters, between the different handwritten digits. Yeah, it's basically the same idea. You're just connecting the data over time. We also used it, for instance, for showing neural network training. So what we've done is we've taken a classification problem, an image classification problem again, and what people use is this confusion matrix. So it basically says which classes are confused by the network. And if you take this information as a feature vector and you project it, this is what you get. So here, that where we have this star here, that would be the perfect classification. But also, if you see every pass is a training, is one training that was started, you also see that some of the trained models are actually moving pretty close to the perfect classification. And in this paper, we also tried to characterize those patterns, so those trajectory patterns for single states per curve and also multiple states per curve. So you could have bundles, you could have similar shapes that appear, and they all have a different interpretation. And we also started to use this technique to project how people make use of our interactive tools. So for instance, what you do here is you see your tool as a feature vector with all the features it has where a user can click on. And then every time the user clicks, you just project this. And there you can then see interesting patterns appear and how people use your tool. So we try to apply it to various kinds of problems. This would be then where we ran a user study where you see then patterns appear how people are using your tool. And this is quite helpful to understand how people then make use, which features are like used more in which kind of order and so on. And so you could have this embedding driven layout. This is what I've shown to you, but you could also show the history of what people do with your tool in a force directed layout. So as a node link diagram. And you can see that both have a value, and you can see some patterns in the force directed layout that you cannot see in the embedding driven layout, and the other way around. And the last example I want to show you is also some recent unpublished work that where we tried to help users in exploring those embeddings. And the idea is that you let users compare clusters appearing in the embedding and give them some means to compare those clusters. And I just need 3 or 4 more minutes to explain that. The first puzzle piece is that you need to be able to represent items and group relationships. So we talked about item to item relationships, those trajectories. Showing items to groups is also not too difficult. So you have items and you just connect them to the groups they belong to. You can have groups to items. So if you have items, you can show to which groups, so to show which items belong to a certain group. And you can show group to group relationships. And this is what we need in order to let people explore those embeddings. And the second puzzle piece is then that the tool needs to be able to show summary and difference visualizations. So for instance, for the chessboard, it's easy. If you compare 2 single states, you can just highlight the parts that have been changed. For the Rubiks Cube, it's the same. If you have a chemical example, you could show the maximum common substructure. So it's the difference between those two. And if you have general high dimensional data, you can use box plots and other representations to let the user know which attributes actually characterize a cluster and which one are different. And this would be then if you have a group of chessboards that the user selected, then of course also the difference visualization looks more nuanced, right? Also for the chemical compound thing. And this would be this example. You again have this projection, and users can interactively create those clusters and then connect those clusters, and they're able to follow, basically to compare what's different between all states between F and G. And then we are having the summary visualization and the difference visualization that is explaining the difference between those. And that works for chess, but it also works for chemical compounds or for gene expression, for instance. This is applied to gene expression data. You have different clusters with different tumor types, for instance, and the tool is trying to explain what's the difference between patients in this and in that group. And it's showing, for instance, which genes are characterizing this group and which genes are making those groups different to each other. And this brings me to the end already. We applied this also, or we integrated this also in this cohort analysis tool, where you can then also use this projection of this cell line or tissue cohorts and compare what makes the clusters in the projection different to each other. This brings me to the end of my presentation. And in the last about 15 minutes, I've showed you some of our projects where visualization and visual analytics is used for exploring the data, for confirming patterns that you or others are able to find, and also for explaining algorithms and more complex models. And of course, over the last years, I was lucky to work with many brilliant students and colleagues. So I want to use the opportunity also to thank everyone who contributed to the results. Well, thanks for joining and for taking the time to watch the talk. Thank you.
Thank you very much. That was really interesting. So if anyone has any questions, please feel free to type them into the chat window or the Q&A to get the ball rolling. So I am a Rubiks Cube nerd. I have several. I'm not good enough to start speed cubing with the Friedrich Method. I do the layer by layer, the beginner method that you described. Now, in that one, it is, for lack of a better term, it is color agnostic. The color you start with really doesn't matter. You can just pick a color and build from that across. Yeah. In terms of mapping a path, now for if you're doing a speed cube, there is a best color to start with that if you're better than I am, you'll be able to see which color you should start with to get it done with fewer moves. But for beginners, if they are randomly picking a color, does that affect the path or like the pattern that they show, or do all of them, regardless of colors, tend to follow the same path once you visualize it like this?
Yeah, that's a good question. I think it can make a difference, or it will make a difference, but it depends on how you construct the feature vector, right? So if we go back to this is basically the important part. It depends on how you transform your Rubik's Cube into a feature vector, and we decided to make this feature vector agnostic to the color, but you could also make it keep the color. But then you would have like different loops probably for different colors that appear. So we decided to make it independent of that, but you could also reconstruct the feature vector in order to see and to investigate if the color makes a difference. That would actually be an interesting thing to try out. Yeah, but we didn't do it. Yeah.
I was just thinking to bring it back to to real data instead of the Rubik's Cube example.
Yes.
The decision of whether to make it agnostic or color dependent. Do you think there would be a likelihood that might mask some piece of information, some pattern that you might miss in a real data set if you made it agnostic? And what would be the best way to try and know if you might be masking it, or would it be best just to visualize both of them and then see what might be there in each?
Yes, unfortunately, the answer is I cannot give you a strategy to come up with the best way to analyze various data sets, because from our experience with what we tried with chess, with gene expression data, with Rubik's Cube, it really makes you can go different. You can go down different paths, and you can try out different things, and you will see different patterns that will tell you or will give you different insights into the problem. Unfortunately, it's in many of the cases not that you create one projection and then like know everything about the problem and the algorithm you're trying to analyze. It's really also that if you play with the hyperparameters, and in the tool we've created, you can actually do that and create a new projection and also compare projections, because sometimes you see something useful in one projection, and that helps you also to interpret the second projection. So it's also that you could have a linked view exploring different projections created for the same data set, but using maybe a different type of feature vector. Maybe you color it differently and so on. So this is where exploration becomes useful, I guess.
All right. Excuse me. Yeah, no, it's. I was afraid that was the answer, because it's getting the data sets and interpreting from them, you always have to run the analyses in different ways and tweak parameters and see what's there. And,
Yes.
you know, I can always hope and cross my fingers that someday someone will find a way to not have to do it. But until then, it's always worth it to do it. So we do have a question here from Arsalan Riaz. He says, first, thanks for the great talk. I'm actually new to the field. Can we visualize cancer progression patterns using genomic data?
I'm not sure if it's meant using these embedded trajectories, but I think it would be worth to be tried. So if you have high-dimensional data that changes over time, it's also, I would be happy to try that out. That's actually an interesting problem. So as part of this collaboration with Boehringer Ingelheim, we have also access to the Genie data set, which is a consortium in the US where they actually have the progression data, but we didn't get so far to try that. But I think it would be like a cool thing to try out, just out of curiosity to see how far you can get using these projections to visualize that. I don't know.
They clarified that they meant using projection space exploration, if that helps expand.
I definitely think that's worth to be tried out, but we haven't had time to do it yet. But I don't see a reason why it wouldn't be potentially useful. And this is always this disclaimer because it's bound to the technique, to the projection you use, and to the hyperparameter. So it could also be that you need to like try different configurations in order to see different aspects and see different potentially interesting things.
All right. So it doesn't look like we have any more questions. I have one question for you, though. In collecting all these data sets and putting it all together and looking at it, what are, in your opinion, the most interesting data sets? Which ones do you always just that make you look at it and go, huh, that's weird?
Yes, so those are always those data sets where you have a personal interest in, right? Where you have a background, where you have, like you love solving Rubiks Cubes, then it's if you have the data set loaded in the tool, it's super fun to explore it. Also, with our colleague who is a professional chess player, of course, they will find obvious things, like you know the different openings, they will spread out at the beginning, but then they can look at strategies. In the tool, you can use coloring, you can use size encoding, so you can take all kinds of meta attributes as well. So for instance, the strength of different players. You can, if you have a gene expression data set, you could use the disease state of a patient, for instance, that you color code on top. And there you always need to make the decision, do I incorporate it into the vector that I project, or do I just want to use it to augment the visualization that I have? And both might give you different results. So long story short, I would think those are the problems where you have a personal interest in, and that could be, it does not need to be a toy data set, it just needs to be a data set where you're currently working on, and you're happy to also load that into our tool. So you just need to bring it into a CSV format that is containing like the sequence, and the columns are basically the high-dimensional vector. Of course, you need to think about the distance metrics then that you apply for the projection, but that's not super difficult, and I think it's fun to play with.